You hear me? Yeah, so let me begin by first thanking the organizers for Hello? Hello? Okay. So let me begin by thanking the organizers for this opportunity to participate in this great meeting, especially Dimitri, who actually helped me a lot in arranging things and so on. So I'm going to tell you something that we have been working in the past few years. Before I begin, I should say that this is not my first time in Brazil. Actually, it's great to be back in Brazil. Let me show you a picture that was taken almost exactly to the date 2 years ago. So I was visiting Witter, and this is at the park. I took the opportunity to take some pictures with capybaras. Okay, so we all know that by now that not all proteins just function by being first folded and then have this binding and so on. So for a long time, actually, people realized that some protein function in a state that's disordered, especially for this structural kind of thing, like for skin, lungs, blood vessel, and so on. So these are elastin protein, they are disordered, and that's the disorder is giving the elasticity that is needed for the biological function. And it really behave like, working like some sort of do we have a pointer here? This is my own, somehow it doesn't work. Wait. No, it's off, okay. Okay, so it really worked like a Fourier entropy spring. Okay, so in the past 15 or 20 years or so, people realized that disordered protein can also function in smarter ways. So they can bind to specific substrates and so on. So these are the IDPs, we call them IDPs. They don't fold spontaneously. They perform permanent function, especially in higher animals like us, for cell regulation, signaling, and so on. And they usually have so-called low complexities. That means they have fewer hydrophobic residues. They have more aromatic, polar, and charged residues. So they don't fold by themselves, but some of them would fold when they are bound. So this is folding upon binding. But not all of them actually even would be substantially ordered when they bind to their substrate. For example, this is an important protein called Sicl. It's very important in the yeast cell cycle. And it functions by having repeated phosphorylation, and the phosphorylation reached the number of 4, 5, or 6, then it would start to bind to this substrate called CDC4. NMR experiments by our collaborators at U of T, Judy Foreman K and others, show that even when it's bound, it is disordered. So people call this a fuzzy complex. And it's also dynamics because there are maybe more than 7 phosphorylation sites, but there's only one binding site. Now recently we realized there could be a secondary binding site, but it's only one main binding site, so it's constantly changing from one binding site to another. And it's remained in a fuzzy disordered state. It's quite expanded. So let me begin by talking about how do people measure the conformational dimensions of disordered protein, and by extension, unfolded protein of globular, and unfolded state of the globular proteins. So 2 methods that people have used, and there are other methods like using NMR and so on, is single molecule FRET and small angle X-ray scattering, SAXS. And there's a controversy that's been long-standing, especially for this particular protein L, and for other proteins as well, is that for people using FRET and using a particular form or interpretation of data, find that the conformational dimension, the rate of gyration of the unfolded state of protein L actually contracts when you lower the denaturant concentration. So it would expand when you add more denaturant. But on the other hand, when people use SAXS, they didn't see much of a change upon the change in denaturant concentration. So these are data that came from a paper from Calvin Plaxco and Tobin Sosnick's group, and summarizing data, previous data. This is from Gilad Haran from Israel, and this is from Bill Eden. So this is a controversy, and so a few years ago with my collaborators, a physicist who is doing FRET at University of Toronto, we decided to look at the formulation, this conventional formulation that people have used to interpret FRET and see whether a resolution can be found by a better understanding of the formulation. I'm going to tell you a bit about this, but the upshot of this is because FRET, usually they put two dyes at the two ends of the chain, and then basically they can only measure one number, which is the average distance between these two dyes. But then they try to generalize it using some formulation so that they can infer a rate of gyration. But if you look at the conformational distribution, I mean, very different end-to-end distance can have the same rate of gyration. This is the gist of it. So I'm going to tell you more about this in subsequent slides. So this is a collaborative work with and he try to use single molecule FRET to measure the dimension of this Sicl protein. This is unphosphorylated. COP means unphosphorylated Sicl. It's 19 residues long. And the first thing is to notice that this A means asphericity. I'm going to define it later. And there's a huge diversity of conformation. This is actually pretty trivial for polymer physicists, but for people interpreting FRET, somehow they miss it for a long time. And people also sometimes have this misconception that if the conformation is strictly symmetric, but if you, I mean, almost none of the conformation is strictly symmetric because unless it's very compact. So this is just to show you if you line up the inertia tensor, I mean, along the principal axis, most of the conformations are not strictly symmetric. So when this asphericity is equal to 0, that means it's totally spherical, and if it's large, the largest number is 1, that means it's rather extended. Okay, so this is just the formal definition of asphericity. So this is really just a very simple simulation. We have a 1 per residues, and then we just have extended volume and simulate all possible conformations. And of course, there's a correlation between the rate of gyration square and the end-to-end distance square, but the spread is pretty big. And if you limit yourself to different asphericity, they're limited to different parts of this distribution. And here is a population distribution of asphericity versus rate of gyration. Okay, so I'm going to tell you how people did this analysis conventionally, but I'm first going to tell you how we do this that we call a sub-ensemble approach, which we think is much more physical. Okay, so with the basic formulation, this is not our formulation. The basic formulation is that the FRET is proportional to this, is a R6 proportionality. So if you have some sort of distribution of the end-to-end distance, you do an integration, you get the average transfer efficiency. Okay, so how are we going to do that? Using the sub-ensemble approach is really quite straightforward. So we first restrict. So we have all the possible conformations. So we just bin them to a sub-ensemble with a narrow range of rate of gyration and a narrow range of asphericity. And then from that, we get a distribution of REE, the end-to-end distance. So you integrate that, then you can get this value, which is really a function, by definition, a function of a narrow range of RG and narrow range of A. And then what we do is we try to match this with the experimental, match this simulation. So in general, you can also have a distribution, but it's hard to infer the distribution independently because basically we just have a number. Later on, we're going to attempt to do that. So the main point here is that given a narrow range of R, G, and A, we get a distribution of REE, the end-to-end distance, then we compute that average transfer efficiency, and then we try to match that with experimental efficiency. The point here is all the conformation that's being considered physical. It's based on a physical chain model. Okay, so here is what I just said. So in our first step, we don't attempt to get a distribution of rate of gyration. In the next step, we're going to do that. So we first leave that open, and then we have this so the experimental transfer efficiency, and then there's this one that's computed by looking at the sub-ensembles, just trying to match them. So the main point is all the conformation that we are using to do this inference are physically possible. Okay, so this is just going to show you the distribution. So here is the REE, and these are different narrow ranges of rate of gyration. These are different values of A, so you get all this distribution of REE, and then we can start doing this procedure. So these are all n equals to 100 residues because the Sicl is 90 residues, and then we put in 10 residues to account for the extra length of the linker to the dies. Okay, so that's what we do. What we do. So how did other people do it? So there are two main methods. They look very different, but they end up with the same kind of result. Sometimes could be quite unphysical. So one is the so-called conventional Gaussian approach. So this basically uses the Gaussian string distribution, but instead of here you have 3 over half the unit bond length. People put in this RG because for the Gaussian chain, the RG, the root mean square RG is equal to 1 over square root 6, the root mean square end-to-end distance. So they put it in. So here you have an REE, and then from the REE you can calculate the average transfer efficiency, and then you try to use RG as a fit parameter. So basically, the best fit parameter for RG, you call that the RG of what you measured, that you infer the RG from the FRET experiments that you conducted. The other is to use the Sanchez mean field theory. It's published in 1979. And then with an extra term, this extra term is to actually, because the Sanchez theory only gives you a distribution of RG, and Haran decided to use an extra term that basically convert that RG distribution to an REE distribution by assuming that can be given by a distribution of 2 points within a sphere, defined by the RG. Okay, so here is a comparison of these different methods. Okay, so for this is our method, the self-avoiding walk subensemble method. So when the E value changed by that much, the RG changed by that much. So it's actually quite narrow, the range. So if you measure something, measure a transfer efficiency, and going from 0.8 to 0.2, the RG is moving along this line, this dash line. Okay, even if we don't have excluded volume, we can try the same thing without excluded volume. It actually behaves quite similarly. As long as you use this subensemble method, that you actually have a physical conformation that you use to infer rate of gyration. But what happens if you use conventional Gaussian? You see when the FH efficiency changed from 0.8 to 0.2, the spread in RG is much bigger. Well, Sanchez theory, ostensibly, it got excluded volume in it, but so it should be different from the conventional Gaussian, but it gives more or less the same result. You spread out a lot. The reason for this different behavior, so that means if you just have transfer efficiency that you measure, the rate of gyration that you infer actually depends a lot on your polymer model. Okay, so I'm going to come back to say why what the conventional method is problematic. Okay, so this is just an illustration of what we do. So this surface is what we calculate using our subensemble method. So basically, we do a match, and so if this is the measure one, we do a cut, and then we get the difference between the experimental and the predicted, and so along this area within certain experimental tolerance, we get an infer rate of gyration by a subensemble method. Okay, so this is a bit busy, so let's just focus on a few results. So this is for the Sicl result. So these are different inferences. This is this is based on the experimental measurement of single molecule FRET by Guadagno slab, and this is our method of inference. So this area is what is considered to be reasonable by polymer scaling. But if you use the conventional or the Sanchez theory method, actually when the denaturing concentration is high, you actually go way up. That means it's very expanded. But if we use the method that we propose, it's all within bounds. And now you ask the question, what is happening here? What is happening here? Why it go to this region that seems to be unphysical? It's because in the Sanchez theory, you fit this epsilon, this interchain contact energy, and at that point, the signal from the single molecule FRET signal would if you use that formulation, demand that it actually was a strong repulsion. So it's basically trying to force the chain to be very extended. That's the problem, because it assumed that the conformational ensemble is always the same as a homopolymer conformational ensemble. So once the FRET signal is getting smaller, you actually force the whole distribution to expand in order for that particular range of signal within a small end-to-end distance to be small. And so you get into this unphysical situation that the whole conformation is highly extended. But if we use the method that we proposed, this self-avoiding walk subensembles, every conformation is physically reasonable, so this thing would never happen. Okay, look at hydrodynamic radius. So here is a comparison between experiment measured by experiment and then our methods. So this is all very good agreement, but if you use the conventional Gaussian, you also get inferred hydrodynamic radii that are much larger than what is measured by experiment. Okay, so as I said before, so the homogeneous conformational ensemble inferred by conformational Gaussian and Sanchez theory are physically untenable, because if you use the RG as a fitting parameter, what you would find is that when the transfer efficiency is low, you force the conformation to have a very long RG, and if you actually look at it more carefully, that means because the total chain length is fixed, that means in this ensemble that you infer, the persistent length is like 8 residues long, which is highly unphysical. The reason why they got this unphysical result is because they always assume there's a direct match between the REE and the RG, and that match is always based on a homogeneous ensemble of a homopolymer. But of course, proteins as a sequence, the conformational distribution is always heterogeneous, even in a disordered state. So it need not follow this relationship between REE and RG as in homogeneous homopolymer conformational ensemble. Okay, here's more data showing why it's unreasonable. For example, the Haran's prescription of calculating the conditional probability of distribution of REE condition upon a particular RG based on points within a sphere. When the RE is small, it's pretty reasonable. So these blue things are actually simulated using our method. But when the RE is large, you see that it's really a big difference. So this line is what is predicted by this formulation. This is the actual simulated distribution. Okay, so now is the next step. This is more recent work. We try to get a better understanding of this controversy about protein L, the SAXS versus single molecule FRET interpretation. So now we actually try to get a distribution of RG. So a natural thing to do, actually, we could have done this from the beginning, is to get a population distribution of a population distribution as a function of both RG and E, because we can always get a population distribution of RG and REE, and so just using this R to the 6th formula converted to E, so it looks something like that. So what we did before is basically we average in this direction and get the average and drop down, and that gives you this curve, this ROG0 we call the most probable. This is what I just went through before. And now we want to get a distribution of RG condition upon a given value of E, so that means that we are averaging along this direction and also get a distribution, and that is this line. The variation is even more gradual, and this is actually the standard deviation of this distribution. So why are we interested in that? Because we want to know how broad are these, at least baseline distribution, and how similar distribution for different E. So people thought that different E would imply very different RG, but if the distribution, the sort of the baseline distribution of RG overlaps a lot, it means maybe actually we don't have that information. So it's basically this question, to what extent differences in E can be used to discriminate RGs. Okay, so here I use the example of protein L. So there's 2 E value, one is 0.45, one is 0.75. So according to the measurement of Gilad Haran, this is for 7 molar guanidine, this is for 1 molar guanidine. So there's, you can impose any distribution of that because people cannot actually measure the actual distribution, always measure the mean value. It doesn't matter. I mean, we proved that it doesn't matter. So for this, we can use the formulation that I just showed you before to get distribution of RG. They look something like that. So this one is actually for two delta function distribution, one for 0.45, the other 0.75. This is for two broad distribution. They look more or less the same. But the main point here is that for this pair of highly different values of E, you get a baseline distribution of rate of gyration which overlap 75%. So this is the overlapping coefficient. This is sort of an ugly symbol, but it's actually standard in statistics. So this is a more global view of this. If we have two values of transfer efficiency, so how much do the distribution of RG, sort of the zeroth order distribution of RG before you consider any sequence-dependent effect, overlap. So overlaps a lot. So say you have 0.6 and 0.4, one E is 0.4, another E value is 0.6. You have over more than 80% of the distribution, they overlap. Okay, so here's our sort of new interpretation of the protein L results. So these black data points are the same as before. And we use our most probable rate of gyration that I told you before in the first part of my talk. You get the change is more gradual, but still there's a big drop when the denaturing concentration is lower. If you use this, the baseline distribution of RG that I just told you about is more gradual, it's something like this. But still, it's not yet consistent with SAXS because SAXS is like all flat. I mean, this should be more or less constant. So is that possible? That's why I told you about this large overlap. So now I'm just going to propose a scenario that this is possible. In fact, people should have thought about this a long time ago because people look at IDP, they have programs like Ensemble and so on. It's just to get the conformational ensemble that's consistent with all experimental data. So because the same RG can be achieved by different REE values, the ensemble that's consistent with both SAXS and single molecule FRET can always be found, especially when you have this huge overlap. So the point here is, okay, so this is the same plot as before. This is E equals to 0.45, this is 0.74, so it's huge overlap. Say, now if we wish that all this data is consistent with the measure value of RG, the average mean square RG being 25, so it's like this 9. So we just have to restrict. Now this is sort of the baseline distribution, but with the amino acid sequence, we don't know how it actually distributes among themselves. Say if there's certain bias that it restricts to this part of the distribution for 0.45, this is only 1/2 of the distribution, but that means it's only restricting the more intrinsic homopolymer-like conformational distribution to 1/2 of it, just because of the sequence effect. It can achieve the rate of gyration of 25. Okay, now if at lower denaturing concentration, at 1 molar, it gets to 0.74. It's getting harder because now we restrict ourselves to 20% of the distribution, then both of these data would give us the inferred rate of gyration being 25. So what I'm saying is if there's some effect that would actually bring the two ends together, there are actually quite a lot of evidence, maybe not directly for protein L, for other proteins like Elisa Ha has been talking about for a long time, and with no denaturing concentration, the ends tend to come together. Some people may even suspect that maybe the dyes come together. Anyway, so if something like that is happening, but the other part of the chain remains disorder, is entirely possible because only a bias from 50% to 20% that you have different transfer efficiency value measure, but they are consistent with the SAXS data that they have more or less the same rate of gyration. So this work is submitted. If you are interested, you can take a look at this archive preprint. Okay, so that's what all I wanted to say about FRET and SAXS. So here's a summary. So the Sicl data highlights the practical limitation because if the chain length is smaller, the fact that sometimes these traditional way of inferring the rate of gyration being unreasonable is less pronounced, but because Σ1 is longer, is 90, if you use the traditional conventional method, you can get clearly unphysical result. So another important point should be obvious, is these order state of proteins are heterogeneous, conformational ensemble. They are not adequately represented by this homogeneously expanded or contracted conformational ensemble, as assumed by the conventional methods of inference. So just re-emphasize that the sub-ensemble self-avoiding walk method is based on explicit chain model. It limits all our consideration to chain conformation that are physically accessible. So it should give us physical results rather than sometimes give us unphysical result. So a single value of experimental transfer efficiency is generally consistent with highly diverse multiple values of rate of gyration. So the conditional distribution of rate of gyration conditional of a particular value of E is very broad, and even the E values are very different. They overlap a lot. So there's a ample opportunity for these things to be consistent, like in the protein case, rather than if we use the conventional method, always give you a particular value of RG, and that would give us inconsistency in terms of the interpretation. So let's now switch gear a bit and talk about this liquid-liquid phase separation of intrinsically disordered proteins and membraneless organelles. Okay, so this is even more recent from an experimental point of view, because for the IDP having some smart functional roles, people have known this for 15, 20 years by now. But in the past few years, people realize that IDP may not only just act individually, they can actually come together and form this liquid-like, gel-like, liquid-like thing that's reversible. This is not like amyloids. And this turned out to be very important for biological functions. So people call them assemblage, and oftentimes they got RNA molecules in it as well. So we're trying to get some physical insight into how this phase separation occurs. So, okay, so just some background. The last talk from yesterday was talk about these synapses, and actually this IDP phase separation is important for that as well. So this is two proteins that are the prominent components. This is not my work. This is a work coming out from Hong Kong for what people call the postsynaptic densities. So these are really these liquid-like droplets composed of these. The main component is two kinds of proteins. And mutation of these proteins, these people found that even in vitro, they would actually make it less likely to phase separate. And those mutations are related to autism. And those mutations related to autism in humans, and also they can lock out those genes, and they also see autistic kind of behavior for mice. Okay, so this is an example to show that liquid-liquid phase-like phase separation for IDPs, if they don't perform properly, it can have pathological consequences. Okay, so organelles are literally little organs in the cell, so we are all familiar with mitochondria, chloroplasts. These are all with membranes, but there are also liquid-like droplets in the cell that serve important function in that in temporally and then spatially, different reaction can happen at different places. And sometimes these are called membraneless organelles. Here are a few examples. These P granules, liquid droplets. They are liquid and they are reversible because people now use this commonly used technique. Is they fluorescent recovery after photobleaching. That means they photobleach something, the droplet, and then after a while, because the proteins are also on the outside, they could diffuse in so it can recover. Nucleolus is part of the eukaryote nucleus. This recent paper coming out of Princeton showing that it largely can be viewed as a coexistence of several different liquid phases with different components of proteins and RNA and so on. Okay, so we are going to focus more on this particular protein. We call it DDX4, RNA helicase DDX4, they form these membranous organelles in the cell, and they also can form, I mean, these experiments done a few years ago in Toronto show that it can also form in vitro. So it's important in a whole bunch of biological functions, these kind of membranous organelles. Interesting thing is that the single-stranded DNA would partition into this droplet, whereas double-stranded DNA don't. So the method that the experimentalists use is to put in a fluorescent protein there, and then you can actually look under the microscope to see these DDX4 actually from droplets, and these are just controls showing that these droplets are different from other droplets that people have already known. Okay, so here are the experimental results. So this is the fluorescent recovery after photobleaching. So it's clearly a liquid-like thing that the DDX4 molecules can diffuse in and diffuse out, both in live cells and also in vitro. So these are actual pictures for the cells, and if you lower the temperature, these droplets starts to form. And you can also trigger the droplets to form by lowering ionic strength. So presumably, because these things are driven by electrostatic interaction, so if the ionic strength is lower from 150 millimolar NaCl to a much lower ionic strength, this thing would start to form. And in vitro, we can also trigger it to form by lowering the temperature from 50 degrees Celsius down to 22, and then you raise the temperature again, they would dissolve. So these are proteins, so their behavior depends on the amino acid sequence. So the experimentalists also looked at that, and so these are the distribution of the charges. Blue is negative, red is positive. So it has this block-like kind of pattern. And they consider a mutant, which is, they call it CS, is a charge scramble mutants of this protein, wherein the charges are more spread out. And another mutant that they consider is to change 9 of the phenylalanine to alanine. Okay, so this wild type protein phase separate, this doesn't, and this also doesn't. So the charge pattern is important for this phase separation, and also presumably the aromatic interaction is also important. It's not really a hydrophobic effect because both of these are hydrophobic, so most likely it's some sort of a cation pi interaction that's at work. Okay, so we are trying to get a handle on how this charge pattern affects phase separation. So we adopted a simplified theory, but it's one step beyond the usual mean field Fourier-Huygens or overbit one theory. It can take care of the sequence dependent in an approximate way. So people call this a random phase approximation. I think the random phase approximation at first was used by Bohm and Pines in looking at electron gas and so on. But DeGene somehow thought that this is similar to that. He didn't explain exactly how, but it's in his 1979 book. He said this is like the random phase approximation. So I think ever since then, people just look at this theory with a quadratic approximation. They call them random phase approximation in polymer physics. So we are all physicists, so we are familiar with this phase diagram. So as I said, RPA accounts for pairwise electrostatic interaction by neglecting higher order density correlation. So these are the charge densities. So here just the outline of the ingredients. It has a Fourier-Huygens excluded volume part, which is this the standard Fourier-Huygens term. This A is just the size of this lattice, the linear dimension of it. These phi's are the volume fraction. Phi m is for the polymer, phi c is for the counter ion, and phi s is for the salt. And this is the RPA term for the electrostatic free energy. It can be simplified into this form, and whereas this G matrix looks something like this. So what it turns out is that after some algebraic simplification, you can just input the charge sequence and then get a phase behavior out of this theory. So we apply it to a whole bunch of toy sequences, as well as the wild type DDX4 and also the charge scramble DDX4. These are 236 residues long. These are shorter or longer. Okay, so we just use the standard method to do this phase separation. So you got the free energy as a function of the volume fraction. So the coexistence point, you get a common tangent, and then for the spin-nodal curve is determined by the zeros of the second derivative of this free energy function. So this is pretty standard for physicists. So this is what we got. This is our coexistence curve. This is the reduced temperature. So actually, in hindsight, it's pretty obvious when you have the sequences have long blocks of positive and then long blocks of negative, then they tend to phase separate more. So this corresponds to these things. If they are all sort of like strictly alternating, then it's much harder to phase separate. So now we apply this to the DDX4 sequence, the wild type, and also the charge scramble mutants. So these are the patterns. I think I showed you this before. So this is the wild type DDX4 according to our RPA theory that we constructed. This is the charge scramble. So clearly, the critical point of this is much higher than that. This would phase separate much more easily than this one. So we can also look at the source dependent using the same theory. We don't use actually the Debye screening because the counter ions are actually explicitly treated in this theory. We also got this behavior that is quite similar to what is observed by experiment. But I just show you the trend in terms of the reduced temperature. That's pretty good already, I think, for a simplified theory. But we so go a step further to try to match the temperature. So this involves even more approximation because at least so far, I mean, we are trying hard on it. I think Keynes is also doing this too. It's still because the long-range interaction, the long-range Coulomb interaction work quite well in this RPA, but short-range interaction still requires some work. So we, at least at this point, we can actually put in the sequence of aromatic and these sort of things into it. So instead, we just introduce an extra Fourier-Huggins term like so, and basically it's another fitting parameter to account for the possible cation pi interaction and pi-pi interactions. And so we have two more parameters to fit, and then we fit it to the, so these are the data points from experiment, these are fitting curve, and so we can now fit to a real temperature in Celsius. Once we achieve this fit by using the sort dependence data, we can do the phase diagram. Oh, by the way, these are the dotted lines are the spin-nodal boundaries. These are the coexistence boundary. So we found that DDX4, the wild type, phase separate. And in this set of fitting parameter, the charge scramble, the critical point is almost to 0 degrees Celsius. So it's consistent with the experimental observations that the charge scramble DDX4 doesn't phase separate. Okay, just to show you a picture taken by our experimental collaborator showing that spin-nodal decomposition also occurs for these DDX4 proteins as well. Here, I think it's just by drying, at this end, it's just drying very quickly. I mean, the concentration is getting higher and higher, and then the whole thing have the typical patterns of spin-nodal decomposition. Okay, so the next step that we try is to look at a possible relationship between single chain behavior and multiple chain phase separation. So this is based on a set of toy sequences studied by Rohit Papoor's group in Washington University in St. Louis in this paper in PNAS a few years ago. So these are toy sequences that he simulated using an implicit solvent but all atom model. It shows that, so for this very blocky, basically it's just a die-block sequence, just all negative, all positive. The rate of gyration is smaller, it's this one. And then when this straightly alternating, then the rate of gyration is bigger. And Rohit correlated this with a parameter that he proposed. It's called kappa, it's some sort of a charge pattern parameter that I'm going to define for you later on. It's some sort of a correlation, a reasonable correlation between this parameter and how large or how small the rate of gyration is. Okay, so the thing that we do is that we look at this and ask, oh, now that we have this RPA, which is quite easy to use because it involves a bit of numerical calculation to get the the critical point and so on, but it's not a simulation, so it can be done quite quickly. So we apply RPA to these 30 sequences, and here are the phase diagrams, and we can get the critical point and plot the critical point, the log of the critical point versus the simulated RG done by Rohit and his co-workers. So it's a really quite good correlation, and it's very sharp. So there's I think a change of RG of at most double or less, 50%, but the change in this critical temperature is almost 50 times, more than 50 times, and it obeys some sort of a scaling like this, but don't take this too seriously because it will change the prescription for calculating the self-energy and so on. This could change, but anyway, it's a sharp dependence. The critical point actually depends. The multiple chain phase separation critical point actually vary quite sharply with the single chain rate of gyration. So as I alluded to before several times, in hindsight, this is all pretty intuitive and obvious because if you have die block, the whole block of positive and negative can come together. Of course, that would make the chain more compact because it's a track and also making it easier to phase separate. And if you have something like strictly alternating charge, positive and negative charge, then the attraction is weaker, and so the rate of gyration is not going to be that small. It's going to be more extended. At the same time, it's also harder to phase separate. So I told you about this kappa parameter that Papu proposed. It's basically charge asymmetry factor, this sigma. So then he takes a window of this with 5 or 6 residue, which is sliding along the sequence, and compare that with the overall charge asymmetry and get this kappa. So so you can see that if you are positive, negative, so that means this asymmetry will be big all along, so we get a higher kappa. Another one is SCD, is called a sequence charge decoration. Actually, is proposed by Professor Gauss here in the audience, and based on a variation theory. This is also a charge pattern parameter. But we found out that this is better because it takes care of non-local effect along the sequence, because this is always just sliding along the sequence, so it's basically all local. But this is actually summing over all I and j by this square root j minus I factor. I think it's coming out from the Gaussian chain basis of the theory. Okay, so we can compare these two charge pattern parameter. Say for RG, this RG is actually simulated. This is not from theory. It's not from analytical theory. It's simulated by Papu's group. So as I showed you before, these green lines are the function of kappa. So they show a reasonable good correlation, but at the same time there's more fluctuation. But we use SCD, actually it's much smoother. Now for the multiple chain phase separation, this critical temperature actually is almost perfect correlation with SCD, but for kappa is more scattered. I don't know how fundamental is this. I mean, this is I found this actually more impressive because RG is actually simulated. It's not from theory, but it's correlated very well with this theoretical parameter. Here, perhaps there's still some sort of underlying relationship between RPA and King's theory, because both are at a certain level based on the Gaussian chain model. But anyway, it seems that SCD captured the behavior of the trend better than kappa. Okay, so these are more recent work that we hope to submit in a few weeks. So I told you before that, like in the nucleolus, there are various coexistent phases. So it would be interesting to find out. I mean, of course, this is very rudimentary. The real membranous organelles are very complicated with hundreds of kinds of proteins and nucleic acid and so on. But is there something that we can learn from this simple theory using RPA to see whether there's some kind of fuzzy molecular recognition? Because you think that in the cell there's all kinds of IDP. How come they don't all just come together in some sort of a glue, right? So this is our maybe feeble attempt to take a look at this. So we extend the RPA to two sequences instead of one sequence. So the extension is pretty simple. So now you, instead of just one phi for the polymer, we have phi 1 and phi 2, and this matrix is just extended to this matrix. Is this a Green's function between 1 and 1, 2 and 2, 1 and 2, and 2 and 1. Because the RPA as an approximation is based on a Gaussian chain model, so you don't have correlation. The cross terms are 0 in the approximation. So basically, you have these things. And then you just plug this in, and this sigma 1 and sigma 2s are the sequence of charges on these two sequences. So again, we can now calculate the free energy in RPA and then use the simple method, the usual method to identify these dotted lines that are spinodal boundaries, and then the coexistence boundary can be found by actually calculating when you have a point about volume fraction, you figure out whether they're phase separated volume fraction that will have a lower free energy. So here are the results. So we look at 6 pair of sequences. This one is always there, but there's different sequences on the bottom. And we look at the difference in SCD. So these are actually pretty similar. The SCD, the minus SCD are pretty similar. This is very different. And their phase boundary, their phase behavior are also very different. So what this means is that this timelines means that you start out with about volume fraction here. This is phi 1, this is phi 2. It will phase separate into these 2 points. And they are all kind of having a slope of, well, close to 1, at least in the middle. So that means for these two sequences that are quite similar, both the more dilute phase separated state and the more concentrated phase separated state, the proportion of the two sequence populations are quite similar. But when the two sequences, the patterns of the two sequences are quite different, as characterized by the difference in SCD, then the timeline is not like that. That means in one phase separated state, there's predominantly just one kind of the sequence, and on the other hand, on the other side is the other kinds, the other sequence. So this can be summarized by a plot like this. So this is it's just a simple measure. We use the arctangent so that it's restricted this order parameter from 0 to 1. Basically, it's just something like that. It's the ratio between the volume fraction in one phase separated state versus the other, and it's correlate quite well with the difference in SCDs. Okay, so this is more speculation, but we have been doing this RPA using basically the relative permittivity as a fitting parameter. But we all know that by using Poisson-Boltzmann theory and so on, and we know that inside of a photoprotein, the permittivity is around 4, and in water it's around 80. So if you have an IDP cloud, what is the relative permittivity? Would that affect the electrostatic interaction? So I found out that actually people consider this in what is called effective medium theory, but this is different. This is actually what I thought about, and the results are quite different. And actually in this paper that we are writing up, we're going to address this. The assumptions are quite different from the usual effective medium theory. And here I basically just use this high school method, right? So we have the electric capacitor. Capacitor, we all know how to use Gauss' law to calculate this. And if you think about it, if you just separate them out more or less randomly, the electric capacity of this is the same as this. Based on that, you can get a formula like this. And we can also use the Cauchy-Mazzotti equation from Jackson. We'll study Jackson's. And so this is another formula. Both of them give this kind of behavior. That means even the polymer, the protein volume fraction is just 20%. This thing actually dropped pretty quickly. The electrostatic interaction can be increased quite quickly within the IDP crowd. So this is, I don't think this is correct, but this is just showing you the trend that something like this should be considered. So instead of having a constant relative permittivity, if we use this volume fraction dependent relative permittivity, we get much stronger propensity to phase separate, because effectively it's some sort of a cooperative many-body effect, because when this thing starts to condense, the permittivity is getting lower, and so the electrostatic is getting stronger. So if we have this volume fraction dependent electric relative permittivity, the propensity should be higher than if we just assume that there's a constant relative permittivity. Okay, so here's a brief summary. So membranous organelles are essentially condensed liquid underpinned by IDP liquid-liquid phase separation. Now it seems that it's a fundamental form of cellular organization, so that it can compartmentalize various reactions to different places at different times, and it can also be controlled, because the idea can be there could be a post-translational, like phosphorylation, that sort of thing, can control the formation and dissolution of these membranous organelles. So some IDP phase separation associated with pathology. I told you one example for the synapses, and actually there also work from Tanya Mitek's group in Tennessee, showing that actually many of the amyloid formation is the ITP phase separation, may be some sort of phase separation, may be a precursor to the formation of amyloids. So because these are proteins, this kind of behavior, of course, is dependent on amino acid sequence, and it's a multivariate interaction. So to account for that, using analytical theory, we can't just use mean field approximation. Using mean field approximation, we can get the trend, but then we will have a different parameter for a different sequence. So the attempt that we have been making at least can give you an approximate method to see the trend using the theory to account for the physical effect of different charge patterns. So it seemed to give us a reasonable account, but of course in the long run we have to do some simulation to see what are the limitations of the approximation being used in our PA. And because definitely it's not going to be all correct, it's a gross approximation in there, although there's some success. Okay, so thank you very much for your attention, and the highlighted people are all involved in this work. Is a very able postdoc in my group, Yishen Lin. He did most of the theory, and Jian Fei Song, now he's an associate professor in China, and he did the FRET simulation. And Professor Guadinaru is a FRET guy in our physics department in Toronto, and Julie Foreman K. She does a lot of IDP, NMR, and other studies. Thank you very much.